Today we're going to continue our discussion of Chapter 21 on the living cell. This lecture has 4 parts. The nature and variety of cells and how does a cell work were covered previously. Today we're going to focus on metabolism and cell division. Metabolism is the sum total of all of the chemical reactions that happen in an organism. These chemical reactions are the way in which genes control what happens in an organism, because genes make gene products, proteins, many of which are enzymes that catalyze chemical reactions or facilitate them. Chemical reactions involve energy input and energy release, and so one of the first principles of understanding metabolism is to understand how that energy is carried within and between cells, and that is by the cell's energy currency, which is a molecule called ATP, adenosine triphosphate. Energy is carried from sources of energy to areas of need by this molecule. This molecule is analogous to cash in the human economy because it's a form of currency or money that is usable by all entities in the economy and can be transferred among from sources to areas of need. Adenosine triphosphate is the same way. There are many molecules that carry energy in cells, but ATP is the universal energy carrier that can be used by everyone. It can be used by many different kinds of organisms, and within those organisms it can be used by all of the chemical reactions that need energy input, and chemical reactions that produce energy create ATP so that energy can be used elsewhere. So the function of ATP, how it works, is that it stores energy in a bond between phosphate groups. A phosphate group has a P surrounded by oxygen atoms, and phosphate groups can bind together, and in the bonds between atoms is where energy is stored. In ATP, the usable energy is stored between the second and the third phosphate group. ATP, adenosine triphosphate, the name implies that there are 3 phosphates. Tri means 3, and it's between the second and the third one where the energy can be used when that bond is broken. So removal of a phosphate group provides energy to a chemical reaction. If energy needs to be stored, we add that phosphate group back, and that energy is stored in that chemical bond. Here is the molecule ATP, and notice the 3 phosphate groups on the right. It's between that second one and the third one where the usable energy is stored. Now, another key question in metabolism is, where does the energy come from in the first place? ATP can be used to transfer energy within cells, between cells, but something I haven't mentioned yet is that during the course of chemical reactions happening, heat is also produced, which is a form of energy, and that means energy is lost from organisms during metabolism. ATP is moved around, but energy is lost. So that energy needs to be replaced. Well, ultimately, what is the source of all energy on Earth? Where does that energy come from? It's from sunlight. And that energy is brought into our ecosystems and made usable for organisms by a key chemical reaction called photosynthesis. Now, how do phototrophs, which are those organisms that use sunlight as sources of energy, how do they get energy from light? Photosynthesis is the chemical reaction. Specifically, what happens is that pigments in chloroplasts absorb red and blue light. That's why those pigments look green to you, is because green light is being reflected, red and blue is being absorbed. And in the pigments, electrons are excited. The energy of these excited electrons is captured or harnessed and used to make ATP, which then can be used to make sugars, and sugars can be used to build structures, or they can be broken down to make ATP to power other chemical reactions. But ultimately, this is, in a nutshell, what photosynthesis does. Pigments capture the light energy, those excited electrons are used to make ATP, which is used to make sugars, and then sugars carry energy that can then be broken down to make more ATP elsewhere in a cell. Overall, the net chemical reaction of photosynthesis is energy input from sunlight plus carbon dioxide from the air plus water results in carbohydrates or sugars plus oxygen, molecular oxygen, as a byproduct. So, why are leaves green? Because they're reflecting green light and absorbing red and blue light in their pigments of the chloroplasts, which are the organelles where photosynthesis is taking place. Here is a diagram of photosynthesis showing the input of carbon dioxide and the formation of a sugar molecule. What's shown here is glucose. That is a ring compound, and it is the simple sugar that is formed in photosynthesis in a leaf. Okay, so let's say a plant has made sugar from photosynthesis. Now, how does it go about getting ATP, the universal energy carrier, out of those sugars? Or let's say that an animal has consumed some sugars, eaten a piece of fruit. How does an animal go about getting ATP out of those sugars? ATP is the universal energy currency, but sugars are also really important for carrying energy within and between organisms. In order to extract energy from sugars, it needs to be converted into ATP. The first step in this process is a chemical reaction called glycolysis. Glyco refers to the sugar. Lysis is breaking down or destroying. So we're going to break down the sugar, at least partly. And glycolysis first splits the glucose molecule, and the result is two molecules of something called pyruvic acid, plus you get two ATP molecules and two molecules of NADH. NADH is an energy carrier. It carries electrons, and that later can be used to help make more ATP. So we get pyruvic acid, which still contains a lot of energy. We're not done with that yet, but we do get two ATPs at this point and two NADHs, which we can use. So one molecule of glucose makes 6 to 8 ATP from glycolysis. This is because we get 2 ATP here plus 2 NADH that can be used to make ATP. This is a diagram of glycolysis. You don't need to memorize the details of this, but I want you to see at the top, the glucose molecule, there is a little bit of ATP input. This is a good example of a chemical reaction that ultimately is producing energy, producing ATP, but it needs energy input at the beginning to get it going. If we break down glucose into two molecules of pyruvic acid, which are at the bottom, we first need the input of 2 ATPs. Then, in the further steps of the chemical reaction, 2 NADH molecules are released and 2 plus 2, or 4 ATP molecules are released. So, I mentioned that we're not done with that pyruvic acid. Pyruvic acid is one of the byproducts of glycolysis, and lots of energy can still be extracted from the pyruvic acid. We don't just end up with those small numbers of ATP that we mentioned on the previous slide. In the presence of oxygen, a really efficient way to create more ATP from pyruvic acid is via cellular respiration. Respiration has two parts. First of all, the Krebs cycle, where pyruvic acid is further broken down. Carbon dioxide is produced, and a little bit more ATP and NADH are generated. But then, what makes most of the ATP is the electron transport chain. Lots of ATP is produced as electrons are passed from NADH to oxygen. Oxygen is a strong electron acceptor or attractor, and that attraction of those electrons to oxygen is powerful, and it drives the production of a whole bunch of ATP. In addition, water is produced as a byproduct. This is why when you breathe out, you're breathing out a lot of water vapor and carbon dioxide. These are the byproducts of all the cellular respiration going on in your cells. Now, ask yourself a minute, what is the organelle in your cells where respiration is happening? We just talked about this in the previous lecture. That's right, that is the mitochondria. The result of this process is 36 to 38 ATP, depending on the details. And most of that is from the electron transport chain. This is a pretty neat picture of, I think, an elk expelling water vapor during respiration. This is breathing out just like you do and showing the water vapor that is being expelled. And what you should also keep in mind is that there's a lot of carbon dioxide in that same breath emitting from that animal. Now, a lot of organisms live in habitats that don't have a lot of oxygen. Can you think of any habitats without oxygen or with limited oxygen? One of them is sitting there with you, that is your gut. Your large intestine does not have very much oxygen at all, and there are lots of bacteria living in your gut. They need to extract as much energy as they can from the molecules presented to them, and they need to do that without oxygen, without that powerful electron acceptor to power the production of a whole bunch of ATP. The alternative to that is fermentation. One alternative to that is fermentation. Fermentation is a way to get some ATP from pyruvic acid without oxygen. Pyruvic acid, remember, is the product of glycolysis, that first step. So with respiration, you start with glycolysis, and then you have the Krebs cycle and the electron transport chain generating a lot of ATP. With fermentation, we're going to start with glycolysis, from which we get pyruvic acid, but then we're going to extract only just a little bit more ATP by converting pyruvic acid into a byproduct. The byproduct has less energy than pyruvic acid, and we're taking that energy out and making ATP. Now, it's not as efficient as respiration, and so what we end up with is an energy-containing molecule that's left over and goes unused. So we have an energy-containing byproduct, indicating the inefficiency of fermentation. Excuse me. One example of an energy-containing byproduct is alcohol. This is produced by yeast when they are fermenting sugars. They also produce carbon dioxide, and as we talked about previously, alcohol and carbon dioxide are key byproducts that we use in food production and beverage production. In animal cells and in some bacteria, another byproduct of fermentation is lactic acid. We discussed previously how some bacteria make a lot of lactic acid, and this is a powerful byproduct. It actually allows them to dominate certain habitats in and on the human body, including female reproductive parts and your large intestine. There are lots of these bacteria that produce lactic acid, and that acid prevents other bacteria and other microbes from growing. We also harness fermentation in food production, as I mentioned a minute ago. Here are some examples: bread, cheese, and wine. Cheeses that are aged are being aged by bacteria and fungi that are exuding enzymes within the cheese, and those enzymes convert the carbohydrates in the cheese into byproducts that are more interesting in flavor, and they make the cheese taste more interesting. Okay, the last part of this chapter is answering the question of how do cells arise from previous cells. The answer is cell division. In eukaryotic organisms, normal cell division, where cells need to be copied, happens all the time. In the growth of any tissue in your body, one cell needs to make 2 copies of itself, and then those cells need to make 2 copies of themselves. This happens by mitosis. Mitosis is a process where cells copy themselves. Their DNA is copied exactly, except when there are little errors made, which does happen occasionally. This is the basic cell division in all eukaryotes. It's not used for sexual reproduction. That's meiosis, which we'll get to in a minute. Remember that the DNA in a normal eukaryotic cell is in a chromosome, which is DNA plus proteins bound together. Haploid cells, and here's a bit of terminology that you need to know. Haploid cells have one copy of each chromosome. Diploid cells have two copies. Most of the cells in your body are diploid. They have two copies of each chromosome. The exception to that is eggs and sperm, which are haploid, and we'll get to those and how those are produced in a moment. Mitosis is a process that copies cells, and it keeps them haploid if they start out haploid, or it keeps them diploid if they're diploid. So a diploid cell that goes through mitosis makes an identical diploid copy of itself. A haploid cell that goes through mitosis makes an identical haploid copy of itself. So the process, in a nutshell, is first the chromosomes are copied, then the chromosomes migrate along spindle fibers, and when that migration happens, step 2, there are two copies of each chromosome, and those copies migrate in opposite directions along these protein fibers into opposite halves of the cell. Once that happens, now we have a copy of each chromosome in each half. Then a nuclear membrane reforms around those new sets of chromosomes, and then the cell splits down the middle, and now we have two copies of each cell. The result is two cells that are identical to the parent cell. This is a diagram of mitosis. At the top, you can see a diploid cell. It has a large chromosome and a small one, and there are two copies of each of those. In the middle, you can see those chromosomes aligning along a central plane, and then the copies of each chromosome, the two copies, have formed at this point. So notice that each copy of each chromosome has formed a sister copy, and it's those sisters, those are identical copies of the same chromosome that are going to migrate into opposite halves of the cell. So at the top, in part A, we have two copies of each chromosome. By the next step, the DNA has been copied again, and now we have four copies of each chromosome. One in the green, two copies in the green X's, and two copies in the purple. In the third step, those extra copies that were just made in step 2 are going to migrate to opposite ends of the cell, and then that cell is going to split in half, and now we have copies of that cell, copies of all the chromosomes in each of the new cells. Here's a diagram of this happening in human cells. Notice the spindle fibers and DNA stained in yellow is the chromosomes that have moved to the opposite ends of each of the cell. Now, meiosis happens in sexual reproduction, only in eukaryotes, and in this process, a diploid cell produces 4 haploid daughter cells that are genetically unique, different than each other. Sexual reproduction, as a result, produces genetically variable offspring. And when this happens in humans, it's a diploid cell that makes haploid sperm or eggs through meiosis. The steps include first copying the chromosomes, and then a unique process to meiosis is crossing over, where there's genetic exchange within pairs of chromosomes, so that the new chromosomes that result are different than either of the ones that started the process. Then we get segregation of doubled chromosomes along spindle fibers into 2 daughter cells, and then segregation and splitting again into 2 new daughter cells from each of those. So we end up with 4 daughter cells, and the result is that those daughter cells have half the normal number of chromosomes. If we started out with a diploid cell, we're going to end up with a haploid cell, 4 haploid cells at the end. This is a diagram of meiosis. At the top, you have 2 copies of each chromosome, so that's a diploid cell. In the next step, each of those copies is doubled or copied, so now we have 4 copies of each chromosome. Those align along the center like in mitosis, but at this stage, now we get crossing over or genetic exchange, pieces moving among the different copies of a chromosome to make unique new chromosomes. Then 2 of each migrate into the opposite halves of that cell along the spindle fibers. That cell splits in half, a new nuclear membrane forms around those new sets of chromosomes, and now we have 2 new cells that each have 2 copies of the chromosome, of each chromosome. Then a similar process happens where those line up along the center, and the copies of each of those chromosomes split from each other and migrate to the opposite ends. The cell pinches in half, nuclear membrane forms, and now at the bottom we only have 1 copy of each chromosome. Compare each of those 4 cells at the bottom to the one at the top. The one at the top has 2 copies of each chromosome, and each of the 4 at the bottom have only 1 copy of each chromosome, so they are haploid. And they're also genetically distinct from one another. This is a zoom in on the crossing over process where pieces of each chromosome exchange among each other when those chromosomes are in tetrad formation, which is when you have 4 copies of a chromosome. You have 2 pairs, all are different copies of the same chromosome. We get crossing over among the adjacent parts of the pairs. This is what results in genetic variation in your eggs and sperm that makes all of your offspring potentially very unique compared to each other and compared to you.